Hi everyone, welcome to Biopsychology Class 1, Faculty of Psychology, Universitas Gadjah Mada. As discussed at the introductory meeting on Biopsychology 1, we will learn about brain activity as a basis for explaining mental processes and human behavior. And this is our reference, Biological Psychology, 13th edition from Kalat. This is our topic for today: neuron and glia, anatomy, neuron, cell structures, neuron type, glia, brain protection, and nutrition. What's inside our brain? Let's take a closer look. The components that make up our brain so that we can experience mental processes such as thinking, remembering, seeing, hearing, dreaming, even falling in love. The nervous system consists of two kinds of cells, neurons and glia. Neurons receive information and transmit it to other cells, when glia serve many functions that we will discuss later. The adult human brain contains approximately 86 billion neurons on average. The exact number varies from person to person. We now take it for granted that the brain is composed of individual cells, but the idea was in doubt as recently as the early 90s. Until then, the best microscopic views revealed little detail about the brain. Observers noted long, thin fibers between one cell body and another, but they could not see whether a fiber merged into the next cell or stopped before it. Neurons and glia in our brain are formed even before we are born. In the womb, since the first 3 weeks of conception, more than 100 billion neurons are formed in the brain and are constantly growing. When the baby is born, some of these neurons die, but others continue to live. In the late 18th, Santiago Ramon's Y. Gadjah used newly developed staining technique to show that a small gap separates the tip of a neuron's fiber from the surface of the next neuron. The brain, like the rest of the body, consists of individual cells. Santiago Ramon y Cajal, a pioneer of neuroscience, is a Spanish investigator, lived in 1852 until 1934. Cajal wanted to become an artist, but his father insisted that he study medicine as a safer way to make a living. He managed to combine the two fields, becoming an outstanding anatomical researcher and illustrator. His detailed drawings of the nervous system are still considered definitely today. Science continues to evolve. We have to thank Gadjah, who discovered the concept of this neuron. In 1892, Gadjah used a new staining technique which he developed from Golgi research. Staining technique is a silver staining technique used to visualize neural tissue under a light microscope, so Gadjah could see that neurons were like individual cells and separated from each other. This understanding is then called the neuron doctrine. It is further known that these individual cells communicate with each other by sending signals to each other. Gadjah received the Nobel Prize for this discovery in 1906. The discovery of Gadjah continues to be carried out by scientists to this day. We can learn more about neurons, the basic building blocks of the neuron system, which are the source of our behavior. Neuron as a cell also have the same structure as other cells in the body. However, this nerve cell has a unit that is different from other cells in terms of its shape. Neurons are specialized cells that function to receive, process, and transmit information to other cells. A collection of neurons will form a nerve. The following image is a visualization of the smallest collection of neuron components consisting of axon, dendrites, forming nerves, and fused nerves, bound by an outer sheath together with blood vessels. The following is an illustration of the neurons in our brain. There is a part in the form of branches like this. There are so many branches here. And then the widen in the middle like this. What is the name of each of these parts? Let's take a closer look. As seen in the previous image, the thick portion here is called the soma or cell body. Then in the center of the soma is the nucleus or cell nucleus. Then there are branches. There are branches called dendrites. Dendrites. The long part is called the axon. This one, the axon, and the tip is called as presynaptic terminal. So let's see more detail. Dendrites are branching fibers that get narrower near their ends. The term dendrite comes from a Greek root word meaning tree. A dendrite branches like a tree. The part of the neuron that receives information. This information is passed on the soma or cell body. Soma, also Greek, means body, contains the nucleus or cell nucleus and the life supporting structure of the cell. So ribosome, mitochondria, most of neurons metabolic work occurs here. The next one is axon. This one is axon. Transmit information to other neurons, organs, or muscle. The term axon also comes from Greek word meaning axis. So the axon conveys an impulse toward other neurons, an organ, or a muscle. Axon can be more than a meter in length, as in the case of axon from your spinal cord to your feet. The last one is presynaptic terminal. This one, presynaptic terminal, the end of the axon, which is the location of the release of chemical substances to transmit information to other neuron. The axons of neurons are shelled by myelin cells. In addition, this form of myelin is insulated so that there are gaps called nodes of Ranvier. This one, see, you can see here. The second function is as a conductor, which speeds up the transmission of information between neurons. Okay, let's take a closer look at the component. Inside the soma, there are several main components, namely plasma membrane, which is the structure that separates the inside and the outside of the cell. Then there is a nucleus or the nuclei of the cell that contain of chromosome. And then there are mitochondria, the green one. Mitochondria, which is responsible for cell metabolic activities, which produce energy for cell activation. Mitochondria also have their own genes separate from the cell nucleus. Furthermore, there are also ribosome. This one, ribosome, like a dot in the black, or parts in charge of synthesizing new protein molecules. Another major component is the endoplasmic reticulum. This one, in a grape one, is responsible for transporting new protein synthesis from the ribosome, while in the presynaptic terminal, it contains synaptic vesicle and neurotransmitter. Okay, don't you remember that the neurons were separated from each other, separated by gaps? And this is a visualization of those gaps, or synapse, where chemicals are released by one neuron and captured by the next. More detailed discussion on neurotransmitters will continue in the next week. So, neurons vary enormously in size, shape, and function. The shape of a neuron determines its connection with other cells and thereby determines its functions. The more branches, the more other neurons are connected. The length of the axon varies. Neurons in the brain have short axons, while neurons in the legs have axons up to almost 1 meter. For example, the widely branching dendrite of the Purkinje cell in the cerebellum, see figure A, enable it to receive input from up to 200,000 other neurons. In the cerebellum, you can imagine in our brain. By contrast, bipolar neurons in the retina, see figure D, have only short branches and sometimes receive input from a few as to other cells. Figure C, this one, contains from portray neurons in the legs that have axon up to almost 1 meter, right? To send the information from the leg to the motor area in the cerebral cortex, right? This one. pyramidal cell of the motor area of the cerebral cortex, and the figure D, yeah, retina, and then figure E, canyon cell from a honeybee. The neurons in our brain are distinguished by their location and function. There are three types, namely sensory neurons, motor neurons, and interneurons. Sensory neurons or afferent neurons. So imagine your hand touching a hot skillet. Sensory neurons in your hands send signal from the sensory organs, namely your hands, to your brain. So you can feel the heat in your hands touching the pen. So from the sensation, bring to the brain. See the arrow. So this is the skin, and they bring it to brain. Furthermore, information about the heat received in your brain will be sent by motor neurons or afferent neurons to your organ muscle, namely your hand against, so that your hands will respond by pulling the hand off the pen. So you can see here the arrow from the brain to the skin, to your hand. Between sensory neurons and motor neurons, in the middle there are interneurons, which function as intermediaries or forwarders of information between the two. These interneurons are slightly different in the shape from motor and sensory neurons. In the interneurons, dendrites and axons are in one structure, you can see here. After studying nerve cells, we will continue to study another component of the nervous system, namely glia or neuroglia. The other components of the nervous system perform many functions. The term glia derived from a Greek word meaning glue. Reflects early investigators' idea that glia were like glue that held the neurons together. Although the concept is obsolete, the term remains. Glia outnumbers neurons in the cerebral cortex, but neurons outnumber glia in several other brain areas, especially the cerebellum. Overall, the numbers are almost equal. According to function and location, the types of glia include astrocytes, microglia, oligodendrocytes, and Schwann cells, and radial glia. Why are they called as astrocytes? Yes, because these glia are shaped like stars. These astrocytes are active patterns of neurons that have function related to axons. These glia enclose the connections between surrounding neurons and protect the neurons from surrounding chemicals. Also pick up the ions and transmitter that the axon releases and releases them back. In this way, astrocytes help synchronize neurons that are functionally interrelated. This allows the axons of these neurons to transmit information in the form of waves. Therefore, astrocytes here play an important role in creating rhythm, such as the rhythm when we breathe. Astrocytes also responsible for dilating blood vessels to carry nutrients to the part of the brain that is being active. According to the tripartite synapse hypothesis, the axon terminals release chemicals that make the surrounding astrocytes also release chemicals that then serve to amplify or modify information that is passed on to nearby neurons. Astrocytes also respond to hormones that affect neurons. How an astrocyte synchronizes associated axons? Branches of the astrocyte in the center surround the presynaptic terminals of related axons. If a few of them are active at once, the astrocyte absorbs some of the chemical they release. It then temporarily inhibits all the axons to which it is connected. When the inhibition ceases, all of the axons are primed to respond again in synchrony. The next type of glia are very small glia cells or microglia. Microglia are part of the immune system. It is in charge of removing viruses and fungi from the brain. Microglia also proliferate after brain damage, removing dead or damaged neurons. Microglia also contribute to learning ability by removing synapses that have become weak. Glia oligodendrocytes are found in the brain and spinal cord, while swan cells are located in the peripheral of the body. These two glia form the myelin sheaths, which surround the axon. Another type of glia is the radial glia. These radial glia are primarily responsible for guiding neural migration during embryonic development. When embryonic development is complete, the radial glia will turn into neurons or other types of glia such as astrocytes or oligodendrocytes. Although the brain needs nutrients from the blood, in reality there are many chemicals that cannot penetrate the brain. This is caused by the blood-brain barrier mechanism. The BBB is a mechanism to prevent the entry of viruses, bacteria, or harmful substance into the brain. Only found in the blood vessels of the brain. BBB inhibits several types of substance so that sometimes complicate the treatment process such as cancer. Most drugs used in chemotherapy fail to penetrate the BBB. Why does this mechanism exist? Why we need a blood-brain barrier? When a virus invades a cell, mechanism within the cell will disrupt the virus particle through the membrane so that the immune system can find the virus. If the cells of the immune system find a virus, they will kill the cell. The next effect, the cell affected by the virus will say, "Hey, immune system, I have a virus. Kill me and save the other cells." This mechanism will work fine in parts of the body where cells can be replaced easily. However, if it occurs in the brain, where the vertebrate brain cannot replace damaged neurons, it can be very dangerous. The mechanism of this BBB includes preventing chemotherapy drugs for cancer or tumor in the brain, thus complicating the treatment process. How the blood-brain barrier works. The blood-brain barrier depends on the endothelial cells that form the walls of the capillaries. Outside the brain, such cells are separated by small gaps, but in the brain, they are joined so tightly that they block viruses, bacteria, and other harmful chemicals from passage. If the blood-brain barrier is such a good defense, you might ask, why don't we have similar walls around all our other organs? The answer is that the barrier keeps out useful chemical as well as harmful chemical. Those useful chemicals include olefins and amino acid, they building blocks for protein. For this chemical to cross the BBB, the brain needs special mechanism not found in the rest of the body. Neurons, especially in vertebrates, depend almost entirely on glucose or sugar. Glucose metabolism itself is in dire need of oxygen. Although the weight of the human brain is only 2% of the total body weight, the brain uses 20% of the oxygen and 25% of the glucose that enters the body. Why are neurons so dependent on glucose? Because glucose is the only nutrient that can penetrate the BBB in large quantities. The body never likes glucose except when starving. The liver produces glucose from all kinds of carbohydrates and amino acids. Before the body can use glucose, the body needs a vitamin B1 or thiamine. Thiamine deficiency, for example, in chronic alcoholism, can cause neural death and lead to Korsakoff syndrome, which is characterized by memory impairment. Okay, this is the end of our discussion of the nerve cell, the basic component of the brain, which is responsible for all of our behavior and mental processes. In the following discussion, we will study how communication between neurons occurs so that information can be transmitted and processed in our brain. Thank you for watching.